All right, so up to this point, we talked about how we're harnessing all the solar energy and we're able to use that energy to make some ATP, make some NADPH. But what about food? Like, we haven't made food yet, right? And the plant, one of the things with photosynthesis is that plants and other producers are able to make their own food. So this is where the Calvin cycle plays a role. So we're actually going to use some of that energy that we got from the ATP and NADPH that we did in the previous step in the light reaction, and actually use that to convert the carbon dioxide into food, right? That's the plant wants to have the food, it wants to store the starch, it wants to use its energy. So let's talk a little bit more about the Calvin cycle reaction. So we can think of the Calvin cycle reaction having basically 3 steps, right? And so this reaction is going to occur in the stroma. So you can see right here when we're looking at the chloroplast right here, the Calvin cycle is going to take place right here, right? So, and it's powered by the ATP and NADPH that we made in the light reaction is going to be used to power this Calvin cycle reaction. And so the end goal here is to actually make the food product. So our end goal is to make glucose because that's the energy source that the cell is going to use. So unlike us who have to consume glucose, producers are able to make their own glucose. So there's 3 steps in this Calvin cycle. The first step is to do the carbon dioxide fixation. The second one is to do the carbon dioxide reduction. And the last step is to be able to start the whole reaction over. We have to regenerate our first substrate. So recall that substrates are the products that are on the left-hand side of the equation. So there are things that can be used to make the final product, right? So we have our substrate here as our what we're going to actually use as we go through the Calvin cycle to eventually make glucose. And then when we say regeneration of the substrate, is that by the end of the cycle, we should again have that same substrate so it can go through another round of reactions. So the first step here is to convert carbon dioxide into an organic molecule. So the first step again is carbon dioxide fixation. So we're going to want to fix this carbon dioxide and convert it into something. So we're going to actually take that carbon dioxide and we're going to attach it to our substrate, which is a 5-carbon sugar. RUBP stands for ribulose 1,5-biphosphate. So what you should recognize from that basically is a 5-carbon molecule that's going to act as an acceptor for carbon dioxide. Recall that glucose is a 6-carbon molecule, right? So you can imagine that you're going to take this carbon dioxide, which is 1 carbon, and you're going to attach it to this ribulose 1,5-biphosphate, RUBP, and we're going to be able to form 1 + 5 into a 6-carbon molecule. This 6-carbon molecule, we're going to do some structural change to it, and we're actually going to split it into 2 3-carbon molecules. But the first step, carbon dioxide fixation, is to basically fix or attach that carbon dioxide to our substrate, which is the RUBP. That's our first step. So now that we've done carbon dioxide fixation, our second step is to do a carbon dioxide reduction. So our goal following carbon dioxide fixation is to take the 3PGA molecules that we have formed and we're going to further reduce them. So we're actually going to gain some electrons. We're going to further reduce them through a series reaction using energy from ATP and NADPH so that we can form G3P, which stands for glyceraldehyde 3-phosphate. So our goal is to actually form this molecule right here. And once we have the G3P, we can actually convert that into glucose. So the purpose of the carbon dioxide reduction is to be able to take the 3PG from the carbon fixation, convert it to G3P, right? So the structural change is changing a little bit using energy that we had made in a light reaction, that now that energy is in electrons that are found in NADPH as well as ATP. and we're going to convert the molecules into G3P, because that G3P has the ability to then be converted downstream into glucose and other organic molecules. So our last step of the Calvin cycle is to actually regenerate that ribulose 1,5-biphosphate, right? So we want to regenerate this RUBP. Why? Because we want to be able to continue doing this Calvin cycle reaction, right? So we want to reform this guy, the RUBP, so that we can then take carbon dioxide, fix the carbon dioxide onto it so that we have the 6-carbon molecule, do some structural changes using ATP and NADPH so that we can eventually form the G3P that will then later be used to make glucose, right? So we have to continue the cycle by being able to make the original substrate to begin with, and once we have the substrate, then we can go in and redo the Calvin cycle again. So by the end of this chapter, you have learned where photosynthesis occurs, and remember they occur only in producer, right? So where does it occur, and then what type of reaction? We have our 2 reactions, if you recall, that we have our photo reaction, and then we have our synthesis reaction, right? So we have the photo that's capturing all the energy, and the synthesis that's harness that energy in order to actually be able to make food for the plant, be able to actually make glucose. Okay, and that's the end for this chapter.